connect to the wireless, it would be a good idea to get that out. We're going to do something in a few minutes. I just need a little bit of help on deciding how your final exam is going to go. So we'll get going. If you do have something that you can connect to Wi-Fi, that would be awesome. So we're going to continue on from the last sort of online lecture and then go talk about some other things that you do need to make sure that you are aware of for the final exam. So again, just to highlight a couple of things as well for you. I think the first thing is the lit review assignment. There is an extension to the Friday, so instead of the Wednesday where it's originally due, I'm going to move that to December the 5th. So just keeping, again, an eye on that. Sorry, and I think I'm going to lose the mic just because it's giving a lot of feedback. And the only other thing to draw your attention to, because I want you to have a good sort of practice for the short answer questions, I am going to be assigning those. You will see like what short answer question you are assigned for the upcoming final exam, sort of as a practice. Instead of having to respond because everyone's getting really busy, I'm just going to evaluate it based on like how clear it is, on whether you've addressed all the points, and it will just be the remainder of your participation mark. You don't have to respond to anyone else's, although I will post some of the best ones as well as some of the other ones that need a little bit of help anonymously so that you can see what your colleagues have been saying about that as well. So our last opt-in quiz continues this week, so that's it. After this week, there are no more opt-in quizzes. This is sort of the last shot at this. And then for your final exam, we will be having a review. It will be done sort of outside of the regular class time. We'll set up a different time. It will be streamed. You'll have access to being able to ask questions, etc. So do you have any questions or anything that you would like to recommend or anything else? Nothing. Okay. All right. So the first thing is I actually wanted to, because we're going to be talking about some of the issues that are going to be directly relevant to today's talk. I don't know if you have ever heard of Ben Barres, who used to be a researcher and professor at the Stanford University. He passed away last December. And one of the things that he passed away from is pancreatic cancer. He died in his 70s. He was a researcher, and one of the things that he was also fundamental in terms of neuroscience, and one of the reasons why he's such a great neuroscientist, beyond the reasons that I'm going to talk about in a few minutes, is that he actually was the first person to identify oligodendrocyte progenitor cells, which are now known as type O2A progenitor cells. Beyond that, while I was at Stanford, one of the things that he was legendary for is his ability to interact with students. And I think that's one of the things that we sort of lose sight of. Like, if you're going to have a really good experience in grad school or whatever you want to do next, it's finding a mentor and a supervisor who's willing to support you in terms of your research, in terms of your ideas. And his lab meetings are legendary. People outside of his lab actually attended his lab meetings because they were so interactive. He would run the entire range and cover all sorts of things related to any field in neuroscience, and they would run 3 or 4 hours, but everyone actually wanted to attend his lab meetings. So if you can, I'm going to ask for a little bit of help. There are some folders up here, and what you're going to do is, if you can, log into this site now. And you're going to help me a little bit on deciding a few things on the next few slides. So once you're all kind of there, I'll give you a few seconds to log on. Not sure how the QR code works, but if you have something on your handheld that can actually directly log on through the QR code, please do so. And Bernice, do you have a sense as to whether or not it's going through, it's recording? Okay, awesome. All right, so let's see if I can Oh, I guess some of you are still doing that. Okay. It's not. I don't think there's supposed to be one. Okay. So, I'm assuming you're all kind of logged in. If you, oh, you're not logged in. Okay. Please work. Okay. Okay. So, regardless of whether you've fully logged on or not, I think the code comes up again on the next slide as well. So, I only need you to pick between A, B, and C. There's no D choice. I decided to make it a little easier. So if you can choose between A, B, and C. And then we'll figure out, based on what you've chosen inside these file folders, what your exam is going to look like. So you can't blame me. It's all on you. Okay, we'll give it a couple of more minutes. Okay, so I think it's sort of stabled out at, no, not yet. Okay, so I think we're kind of stable at about 38% for C. No. Yes, I think C has won. So this is the number of short answer questions that you're going to get on your final exam. No, I didn't tell you that. No. Oh, that's not good. Okay. Do you want to actually see it? Yeah. So this is the number of short answer questions that you've chosen. So do you want to keep that? No. Yes. Okay. So we'll. Yes. Yeah. So we'll use that one as a practice. So you will only get 3 short answer questions on your final exam. You will have 1 short answer where everyone has to write it, and then you'll have a choice of answering any 2 of 3 other ones. So you'll have like question 21, you'll all answer question 21, 22, 23, or 24. You get to pick 2 of those depending on what you want, okay? So you'll only have 3 in total. So you guys are not so great at multiple choice so far. So now we'll go on to sort of the next poll. So you get to do the same thing again. So also file folders A, B, and C. Actually, let's move them around so that they're like this. So we'll just give it a few more seconds. We'll see how you do. No, this is how hard. Yes. Is there an inverse scale? I have no idea. Whatever it is, you guys are deciding. So. Okay, for those of you who are wondering what is actually being pulled here, this is the level of difficulty of your final exam. So choose carefully. Okay, so I think we're all done. So I think the B's have it. Let's start with Let's start with C. So for those of you who picked C, this would have been what your final exam would have been. So no to C. So you picked B. Yes? You guys are good with B, or shall we switch it up? You either have A or B.
Medium or easy, or is it extremely hard? Wait,
Sorry? Yes, it is, kind of, isn't it? Okay, so do we open up B and this is your final answer, or do we switch it up and go with A?
No. Let's just open.
Just open B. Okay. So you're going to have to interpret this one because this is more pictorial, and you'll have to imagine that this is what you're going to be doing. So I'm going to actually assume that you picked A, because that's the answer I know you were all thinking of in your heads, right? I'm hoping that on the final exam you're a little bit better at the multiple choice. You're going to have 20 of the multiple choice. They will be fair, and I'm going to cover them in that review session. But I'm hoping that today's experience with your multiple choice will get you to start thinking really carefully about some of your answers coming up. All right, so let's go back to lecture. So we had sort of talked in the last lecture about PKM zeta and its potential role as the molecule that's involved in long-term engram formation. So this is where you have a long-term memory, usually as a set of synapses that have become strengthened, no longer in the hippocampus, but at the level of the cortex. And this was the first experiment that was done by Todd Sachter's group to demonstrate that PKM zeta was an important factor, and that by inhibiting PKM zeta using a pharmacological inhibitor known as ZIP, that he could actually erase memories. So you learned something, ZIP was given to you, it erased that memory, and it looked like you actually hadn't learned it. And that was the role of PKM zeta, and we talked about it in this context. And again, apologies, I'm not sure this is going to work. We'll see if it does. I'm not holding out for any. Bernice, do you happen to know how to get back to just the arrow? No. Nope. Okay, I'm not sure if that's going to work at all. In any case, one of the things that try the laser. Hit laser again. Oh, maybe. I don't actually see it as a movie thing, though. Okay. Anyways, not important. I'm going to try to find the link. I wasn't able to last time, but I will try to find that link. So if you look at these different dendrites, and then you look at the spines at the ends of the dendrites, and if you were to fill the dendrites so that you could visualize the dendrites and then these little protrusions that are known as spines, and you fill them with GFP, if you were to stain for PKM zeta, this is normally what it looks like. This is PKM zeta, the molecule, the memory molecule, and you can see that it definitely sits inside the dendrite, sitting right under the spines, and usually it's not at the level of the spines. It's ready to go, but it's actually not there. And if we look at how we can activate these different spines, we might see, for example, PKM zeta moving into some of these spines, so it moves into this spine, but it's actually not moved into this spine. So suggesting, just as Todd Sachter had suggested before, PKM zeta is not universally going to go to every last single spine, because spines represent a synapse, and these synapses do not ubiquitously have PKM zeta at every last one of them. Some of them have PKM zeta, so this one has a little bit of PKM zeta, and this one has no PKM zeta. And the same thing here. There's a lot of PKM zeta in this spine, and sitting right beside it, there's another spine that has no PKM zeta at all. So very, very similar to what Todd Sachter had suggested, that potentially this is the memory molecule. Nowadays, though, we're not really satisfied that this is how we should be analyzing the potential role of one of these genes. You needed to know on the last quiz that you took online how or what PKM zeta looks like. So it is a member of the protein kinase C family, and it only contains the catalytically active domain, so it's always on. And if you were to translate the mRNA that goes to every last single spine, then that protein would be constitutively active. However, one of the things that Todd Sachter did not do is what we would traditionally do in life sciences, which is to knock out the gene. So if that gene is so important, let's knock out this gene and let's see what the result of that gene is. So again, there were two groups, and this was again very controversial at the time. Two different groups actually did that exact same thing. They knocked out PKM zeta, and they analyzed what happens to learning and memory in these mice that have no PKM zeta whatsoever. And this is where the controversy arose, and I'm going to take you through some of the controversy right now. So this is from Rick Hugener's paper. And one of the things that he showed, in the wild-type mice, this is the normal structure of what the hippocampus looks like. So you can see the CA1, you can see very clearly the dentate gyrus region. And if you were to look at the knockout animal, so the knockout representing the PKM zeta knockout, one of the things that you would be able to see is that overall the structure, the architecture, the circuitry within the hippocampus for this knockout animal remains more or less the same. So knocking out PKM zeta, you might think has terrible consequences. If it's so ubiquitous, it's so important, knocking out PKM zeta must have really damaged the animal's normal development, but in fact, the animals had nothing wrong with them. The structure of the hippocampus was intact, etc. So again, just to verify that if you were to look at the wild type, and this is sort of the normal PKM zeta, and if you were to look at the knockout and at the same lane, you would see that there is no PKM zeta being expressed. So it is indeed a true knockout. This band is picking up a different sort of gene, and you don't have to worry about it. It's being cross-reactive, and this is the band or the region that you should be looking at, and the knockout animal definitely has no PKM zeta. So structurally, from A, the hippocampus is intact. B, we have confirmed through in situ hybridization that the RNA is no longer being expressed. At the protein level, all of these represent different Western blots down here, the wild type and the heterozygous animal. So again, the heterozygous has a reduced amount of the PKM zeta, and this is the complete knockout. If you were to take a look, PKM zeta, the two identified isoforms, are both completely knocked out in this animal. Because PKM zeta is a member of protein kinase C, and it is a family member of protein kinase C, there are multiple isoforms of protein kinase C. One of the important things to go back, as I'm sure you've learned in first and second year, is to make sure that there is no compensation. You knock something down, or something is missing, does something take its place? Does it upregulate other genes that might compensate for this? And because it is a member of protein kinase C, the logical place to look would be other family members of protein kinase C. So for example, lambda, alpha, beta, delta, mu, etc., all of these remained at the same levels. There was no compensation for every last known family member, including this one here, iota, which I'll get back to a little bit later on. So I'm hoping that you agree that this animal has a normal circuit compared to the wild type, that the gene indeed is knocked out. More importantly, the protein is no longer expressed in these animals within the hippocampus, which is what the protein was taken from. Do you have any questions on this before we go on? So this is just establishing that the knockout animal looks like a normal wild-type animal. The circuitry is intact, but it's just missing that one PKM zeta. There is no other compensation. We're good to go? All right. So we go into electrophysiology, and all of you should now be very familiar with these types of plots. And one of the things that we see, and I'm going to start here down in E, if we take a hippocampal slice and we are doing this type of recording, where are my electrodes based on what you've learned? Where is my stimulating electrode? Where am I delivering this high-frequency stimulation? So I am in the The axons coming off of the CA3, what are those known as? Oh no. Should I be panicking? Just a little bit. Do you remember the Schaffer collaterals? Oh yes, of course. I'm not going to be able to be there sitting beside you during the exam telling you, remember those Schaffer collaterals. So we have the stimulating electrode on the Schaffer collaterals, and what is it making a synaptic connection to, and therefore what neuron am I actually recording from? CA1. So when we do this high frequency stimulation in the open, meaning the wild type animal, just like we've looked at before, and also in the knockout, a couple of key questions. Do we see LTP after high frequency stimulation in the wild type animals, which are these open circles? Bless you. Yes, no, maybe? So you do see LTP, right? So we see the induction, expression, and then the maintenance of LTP. Do we see LTP in the knockout animals, the dark circles? Does it look more or less like the wild type? It sure does. In fact, there is no statistical significant difference. And again, just to make sure that you are reviewing, because your final exam's not that far away, why is there more of a response here? What is actually causing this? What receptor is being expressed more at this synapse? So these are more AMPA receptors, and this is what is happening in both the wild type and knockout animal. So again, to make sure that this is more physiological, and this is a different type of stimulation protocol, we still stimulate the Schaffer collaterals, and we still record from the CA1, but theta burst stimulation, or TBS, theta burst stimulation is a much more physiological type of stimulation protocol than what we would see with high frequency stimulation. So it's just a different way to induce LTP. So instead of doing 100 action potentials per second, I do 100 action potentials for 50 milliseconds, and then I wait a little bit, and then I do another number of action potentials for 50 milliseconds, and it is more like the way in which your brain fires when you're trying to remember something. So in this induction protocol, or how we induce or try to get LTP, does theta burst stimulation also produce LTP in both the knockout and wild type animals? It does. So again, there's no problem here whatsoever right now for either Rick Hugener, who wants to prove that PKM zeta is not important, nor is there any issue right now for Todd Sachter, because LTP is not long-term memory. They are completely separate and distinct. LTP is here in PKM knockouts, not an issue. Here's where things really break down for Todd Sachter, and it is very important that you understand what is going on in this slide. So I'm looking at theta burst stimulation at the top. I'm looking at high frequency down here at the bottom. And there's one additional trace that I'm going to show you in a moment. But let's take a look at what's happening here. So I have the wild type animals in these open circles. Do you remember what ZIP is? So this is the PKM zeta inhibitory peptide, and it is supposed to be very, very specific for PKM zeta. And where you see the black bar, this is where I am applying, or where Rick Hugener's group is applying 4 to 5 micromolar of the ZIP, just like we had seen with Todd Sachter. So Todd Sachter, in that rotating disc paradigm, he had injected ZIP. He saw that memory was erased. So let's take a look at what ZIP is actually really doing. And one of the things that's really surprising about this is, first, before we add ZIP, do we get LTP? Is it higher than what we started with? So this is what we start with. We do theta burst stimulation. Do we get LTP? Yes. Now we add ZIP. And what do you see happening to LTP? LTP is decreasing. Again, nothing wrong with that. So if you're just looking at the open circles, there's nothing wrong whatsoever with the ZIP causing a rundown or the loss of LTP, where it starts off really strongly. As soon as you add ZIP here at the bottom, then LTP starts to nosedive. In fact, you almost get LTD. It's lower than it was before. But here's where things are really bad for Todd Sachter. This is a knockout animal. there is no PKM zeta. In the PKM zeta knockout in these dark circles, should you expect to see a response? Remember, ZIP is supposed to inhibit PKM zeta. So should you see any effect if you have no PKM zeta around? You should not. The expectation, the hypothesis would be that no, there should be no effect. And yet, in the knockout animal, you see exactly the same thing. Very, very troubling. This is incredibly troubling because it looks like if you add ZIP, it's just going to reduce the strength of any synapse within the brain. Okay? So let's take this a little bit further. So again, this is in wild-type animals, and this experiment is only in wild-type animals. And this is actually maybe a worse experiment for Todd Sachter than the one that I just showed you up here at the top. So here are wild-type animals where we do high-frequency stimulation, 100 action potentials per second, and we have two different pathways. And again, you need to go back to what I talked to you about before. One of those pathways is the Schaffer collaterals. They make direct synaptic connections to the CA1 neurons. And the other one, the other pathway that loops around to the other hippocampus, the opposite side, what were those called? Those are commissural fibers. And so if we tetanize the commissural fibers, which make no synapses, you shouldn't get strengthening. So you do both. You have two different electrodes on two different groups of axons. On one, on the Schaffers, we get strengthening. On the other one, of course you're not going to get strengthening because the axon that's running off to the opposite hippocampus isn't making connections. But also equally troubling, when I add ZIP here to a non-stimulated, just a normal baseline synapse, adding ZIP here to this non-associated, non-LTP'd synapse also causes a reduction. So this is a very, very non-specific effect. And again, it doesn't matter if you're doing it in wild type or if you're doing it in knockout, the results are always the same. When you add ZIP, whether it is to the expected strengthened pathway or just to a control pathway where it's not even making synaptic connections, when you add ZIP, even if no PKM zeta is around, it always causes the synapses to get weaker. So do you think ZIP, based on this result, is specific or non-specific? It is a very, very, at least in their hands, a very non-specific effect. Again, to add insult to injury, again, I happen to know because I talked to one of the grad students that were in the lab. One of the reviewers asked them to make sure that they had actually done a number of extra controls. So this figure was added after they had submitted. And one of the things that they were asked to do is, can you use a different inhibitor to show that the effects are actually specific? In other words, PKA is a kinase. It kind of looks like protein kinase C. It's not really the same, but it is sort of a related kinase. And we have a specific inhibitor for protein kinase A called PKI. And when you do exactly the same experiments, just to make sure that your technique is not faulty, to make sure that there's nothing wrong with the animals that you're using, just as a precaution to see if it's not something technically wrong, when they add PKI, where PKA is not supposed to be involved, not supposed to be infected, when you have the strengthened LTP synapse, when you apply PKI, there is no rundown. We'd never see it getting lower. And in the control pathway where it's going to the commissural fiber where there's no connections, also there is no rundown either. So this non-specific inhibitor that we don't expect to see an effect shows us we don't get that effect. So that's great. It's not a technical problem. But what it does mean is that at least in their hands, ZIP is not doing what it's supposed to. It's not supposed to have anything to be able to act on, and yet in wild type or in the knockout animals, it is always causing a reduction of the synaptic response. It is very troubling. It is bad news for Todd Sachter, who has basically built up his entire career over the span of 7 years based on ZIP being a specific inhibitor. And again, keeping in mind that his lab is the primary lab that has been doing most of the experiments, pushing out most of the papers saying that ZIP is involved. Worse news is that if you look at the same issue of Nature in January of 2013, another group, Robert Messing's group, also used behavioral studies to show that long-term memory formation, not LTP, long-term memory formation and learning and memory are also intact in this PKM zeta knockout. They had done separate experiments, they hadn't collaborated, and yet they found exactly the same thing. And again, you don't have to worry about all of the different forms of learning that are tested here, just to show you that several labs in the same year, in the same journal, actually showed that Todd Sachter's hypothesis that PKM zeta was important for learning and memory actually didn't hold true to the hypothesis. It was really a problem with his zip. Any questions on this? Do you yes?
The slide before this one, are all of these experiments done on those commissural fibers?
Are all of the ones up here where we see strengthening done on the commissural fibers?
Yes.
These are the Schaffer collaterals where we have normal synaptic connections. So Schaffer collaterals sit on the same side. They will make synapses. The commissural fibers go to the opposite hippocampus. So we wouldn't expect to see that on the control, which we don't. We don't see strengthening.
Okay,
So the lower traces here are the ones where it's the control pathway. It's not making synaptic connections directly.
okay. And they're just measuring at the same place, they're still measuring?
Yes. Yes. Any other questions? Yes. Preference index on here? Where do you see preference? So they were actually testing a number of different types of memory. So they were looking at condition place preference. They were looking at novel object recognition, all the ones that you would normally examine. All of the behavioral tests for learning as well as memory were perfectly fine in the PKMZ knockout animals. So like place preference would be like condition place preference. There was no differences in the learning and memory.
So then, as soon as they added zip, because I think this is the only one they had zip to,
Yes.
then you see a difference because zip is not specific.
So again, here, zip is not specific. And they came to the same conclusion that zip is actually not working in the ways that they thought it should be working. And again, you can probably make the prediction, even though the legend isn't up there for you, that these animals are all the PKM zeta knockout animals. So you wouldn't expect it to work. There's no PKM zeta, and yet the zip is still working on them. So they came up with exactly the same conclusions. So is Todd Sachter in trouble? Would you work in Todd Sachter's lab? No? But yet people did. And this is one of the individuals that actually went, after this controversy had broken, he actually was really interested in working with Todd Sachter. And one of the things that he actually did was, it took a while. Like for a long period of time, there was complete silence out of the Sachter lab. There was no responses in the journals. There was no rebuttals. It was very unlike the situation with Irisen that we talked about earlier, where Spiegelman came back within like months saying, no, you are wrong. Here's why you are wrong. There was complete silence from Sachter's lab for a very long period of time. And then they published in eLife. And in eLife, they actually came up with this really interesting finding that, in fact, Rick Hugener is wrong. So remember, Rick Hugener actually went and checked that there was no compensation. Do you remember that Western blot where he looked at all these different PKC isoforms? And he was very careful, like none of the other protein kinase C isoforms were actually changing. But Todd Sachter is actually coming back and saying, actually, you're wrong, because there is compensation by a different form of PKM zeta. And again, I'm not going to ask you to read through all of this. He calls this the PKC iota. And he was saying that this compensates for the loss of PKM zeta. You just weren't able to pick it up. And there was a lot of controversy about this back and forth. And again, if you were to read through the response, and this is actually the response from Rick Hugener. You can actually go to eLife. eLife keeps track of like everything. You can see the submission date. You can see the reviewer comments. You can see anyone who wanted to comment on it. You can go in and take a look. And Rick Hugener actually went online and commented on this manuscript. And again, he actually comments on this particular finding. So again, you can read through it. He uses some very colorful language. He doesn't actually believe that Todd Sachter is correct here either, but Both of them have decided to drop it for the time being. They're sort of just not talking about it anymore. Rick Kugner felt that he made his point, and Todd Sachter feels that he's kind of been able to rebuff any of his comments. So where are we right now for PKM zeta? If you were asked this on the final exam, you would recognize it as a memory molecule or a potential memory molecule. And you should know some of the experiments that rebuff or speak against whether or not it is a memory molecule or not. And again, just to make sure that you're all aware of it, you should probably be studying that for being able to tell me how you would go back and test for a potential molecule that you would think is involved in learning as well as memory, like PKM zeta. Do you have any other questions before we go on? Yes?
So has no one checked the compensation molecule that he said that?
The iota molecule?
Yeah.
So Rick Kugner says he can't actually see any change.
Okay, but what about
Todd Sachter?
yes, what about him? Like,
Yeah,
does it not
so
look-
Todd Sachter's group, and in that eLife publication, he actually sees a compensation by PKC iota. If you knock out PKM zeta, there's compensation through PKC iota.
But he didn't try to knock out the compensation molecule.
He does. He actually uses an antisense oligonucleotide approach to knock that down to show its effects.
Okay, and then did the effect show that you don't get LTP if there's?
Yes, at least in his hands.
Okay.
Any other questions? Yes.
Sorry, for the traditional neurons, there's no synapse, so zip does not have effect on
No, zip in their hands there. Actually, if you go back to the slide, and maybe I'll ask you to go back to the slide, on the control pathway, the lower pathway, it actually also causes a rundown. If you apply zip, it reduces synaptic strength regardless of whether it has LTP and you have lots of AMPA, or if you have just a few AMPA receptors, it causes a rundown of the response. It doesn't matter. Any synapse actually gets weaker. Any? Yes.
Have they done a PKM zeta and an iota knockout?
A double knockout. I guess if people were really interested in doing that, they could, but I don't think they have. No. It's a good question. Do you not believe in science anymore?
I hate that.
You hate science. Okay. All right. Now that I've turned you all against science, let's go into some of the other things related to science that actually kind of work and is really interesting. So this, by the way, is not Ben Burris. Some of you actually are making good use of this individual's products. Actually, not as many as there used to be. So if you have an Apple, do you know who this is? Steve Jobs. Right. So it is Steve Jobs. Okay. So I'm going to talk to you a little bit about the placebo effect, why there's a neurobiological basis for the placebo effect. It is a real effect. It is a very powerful effect, and how we can actually measure this. So a key question, because this is how I'm going to end today's lecture. The placebo effect, you have to believe something is true, right? So this pill is going to make me better. More or less what the placebo effect is. Do you understand the placebo effect in a different way? No? So there has to be belief that something that, even if there's nothing active in it, it's just a sugar pill, it will help you. And if you believe strong enough in that, it will help you. Correct? So do you think we can actually see the placebo effect in animals? Can you fool an animal and make it believe in something so that it can actually exhibit a placebo effect? And that's a key question. I'm going to leave you with that question. You're going to need to know that for the final, and I'll take that up in the next class. So one of the reasons why I'm particularly interested in this, so if you look at Steve Jobs and you look at Ben Burris, one of the things that you will know is both of them passed away from pancreatic cancer. Ben Burris, typical form of pancreatic cancer, he was in his 70s. And as you get older, unfortunately, pancreatic cancer is one of those types of cancers that it is almost inevitable you will, as you get older, the risk for developing pancreatic cancer increases. It is actually a very common form of cancer. It's the fourth most common form of cancer that develops. And sad to say, as you get into your 60s and 70s, the likelihood that you're going to get pancreatic cancer goes up. In fact, what makes this type of cancer even worse, until you're at a stage where it becomes really debilitating and you can't be helped, it remains pretty silent. You won't actually have any of the effects of having this particular cancer. And then suddenly, one day, you'll have a really rapid decline in your weight. You'll have this really unexpected weight loss. And then suddenly, because now the cancer is like really big, it's starting to affect some of the ductal cells, you'll actually start exhibiting pain in the upper abdominal region, and then people start to wonder what's going on. So again, as this disease progresses, and it progresses incredibly quickly, you will also have jaundice or yellow discoloration of the skin. You will also have hypercoagulability. There are different things that are going to cause your blood to clot much more than it normally should, that hypercoagulation. And because of that jaundice, because it's affecting your liver system and it's affecting a variety of other things, you're also going to have steatorrhea, or you're going to have lots of lipids within your stool. So there are a lot of things. And again, most of these, as you get older, you encounter some of them, so you always kind of wonder, like, is this a form of cancer or is this something else? And the reason why this is so sad is that Steve Jobs actually had an uncommon or atypical form of pancreatic cancer. This is, again, as you get older, Ben Barres, he was diagnosed with it. He was at Stanford University. Steve Jobs was just up the street in Palo Alto. So Palo Alto is part of the Stanford University community, and it's one of the best healthcare facilities in the entire world. And Steve Jobs is one of the richest people in the world, and he goes to Stanford University, and he gets diagnosed with an operable form of pancreatic cancer. If you happen to read Walter Isaacson's book, where he outlines Steve Jobs' life and his death, the attending physician who had been really smart, picked up on the fact that maybe he has something really serious, decides to do an ultrasound. He sees a little shadow on his pancreatic duct. He decides to investigate it further by doing a biopsy. None of this would occur, by the way, for any of you, because you just don't have that kind of money. Steve Jobs does. He's in this really high technology focused university healthcare center, and he has all of these tests done, and the physician breaks down, cries, and says, "Thank God you have an operable form of pancreatic cancer, and we caught it early." So what does Steve Jobs decide to do? He left it. So he decided that, you know what, I'm going to try to cure my cancer based on what my own belief system is, which is like a very different type of antioxidant-enriched diet. I'm going to listen to my dietitian. And even though you're telling me that I can get rid of this particular form of cancer, and even though I have the money to pay for this operation, and even though the expectancy for my life, if I had this done early, would be another 50 years, he decides to ignore all of that advice, and he decides to go a very, very different route. And again, that's primarily because of his belief system. Again, a couple of other predisposing factors why Steve Jobs was kind of surprised by all of this. He was not at that age. He was only in his 50s. It was very early for him to get this. And again, he was diagnosed because someone really smart picked up on the fact that maybe these signs that he's exhibiting, this weight loss, is actually something more serious. So he wasn't a smoker. Smoking is one of the risk factors that increases the likelihood that you'll get pancreatic cancer. He had a very, very vegan diet, so he had no high fat. He had very low protein in his diet, so diet was not a concern for him. He had not had a Helicobacter pylori infection of the stomach, which is now thought to be a predisposing factor for developing pancreatic cancer. For those of you who have taken health and disease courses, Helicobacter, how many of you, or what percentage of Helicobacter goes undiagnosed? Any ideas? So about a third of this class, actually, you have an infection of Helicobacter. You just don't know that you have an infection of Helicobacter because it's an undiagnosed infection of your gastric lining. So not surprising. You'll know as you get older in life, if you start developing like stomach pains, as you get older. But again, he had none of those issues whatsoever. He had, again, remarkably for Steve Jobs, oh, the alert, okay, nice. It's a test. All right, awesome. Now everyone's checking their cell phones. And those of you who didn't get the message are wondering, am I not important enough to get this message? So again, for Steve Jobs, think about all the things that were going in his favor. Steve Jobs, because he's rich enough and because he's famous enough, had been genotyped fully before all of this had occurred. He had none of the mutations that are normally associated with pancreatic cancer. He had no mutations in BRCA2. He did not have a mutation in the partner and localizer of BRCA2 or PALB. And all of these things were not present in him. So he had been genotyped. He had all of these facilities, but he decided ultimately not to go with a traditional form of treatment. And that makes me very, very angry. So he could have been around for another 50 years, listening to the advice of his doctor, in very good health otherwise, none of these predisposing factors, but he decided ultimately not to do so. Again, a couple of other things to keep in mind. And by the way, even though this isn't neurobiology, I would like you to make sure that you know these last 3 slides related to the biology and the things that cause pancreatic cancer. They will be on your final exam. They will also be on your upcoming quiz this week, so just a little advance warning for you as well. So as you get older, it's more and more likely that you will start developing these small tumors that exist as adenocarcinomas. And it depends on where these adenocarcinomas within the pancreatic system begin. So if it is part of the ductal system that the pancreas has, and again, you don't need to worry about this diagram, but it kind of highlights like the difference of the duct. And this is the pancreas over here. All of the pancreatic juices, of course, are put into the duodenum. And you can have a pancreatic cancer that begins here, or you can have it beginning in a different region of the pancreas. The type that Steve Jobs had, which was completely operable, was known as a neuroendocrine islet tumor. And these tend to be very benign, except if you leave them alone. And if you leave them alone, and your immune system happens to be suppressed because he had a variety of reasons for why he was kind of going to the hospital in the first place, these benign tumors in the region that they're in, they actually started to metastasize to other parts of his body, and he eventually had to get a liver transplant, and eventually he died of other forms of cancer because it had simply spread. So it can be benign, operable, removed, won't spread, but he decided not to do this. Unfortunately for pancreatic cancer, unlike other forms of cancer where we have made tremendous progress in the last 30 years, pancreatic cancer still remains a very poor prognostic indicator. So usually we talk about a 1-year survival rate and a 5-year survival rate. Most cancers now that are caught early, for example, lung cancer or other forms of cancer, the survival rate after 1 year is like 95% or higher, and other forms can be like 98%. Certain forms of leukemia, those types of blood-like cancers, you can have like almost 98% survival after 1 year following diagnosis and treatment. But for pancreatic cancer, it's 50%. It's not very good. So you get diagnosed with pancreatic cancer today, half of those individuals that get diagnosed today won't be alive 1 year from now. After 5 years, it goes all the way down to 20% or lower. So we haven't been able to treat this very well because the pancreas is so essential in you being able to process food as well as sending out different hormones, and it is just really a tough organ system to lose. Again, we have very limited options too. This slide you do not have to memorize. This isn't a slide that I want you to be worried too much about. The surgical options to prolong your life, there is no way to kind of reverse what's going to happen, but to prolong your life as well as to reduce some of the pain, because the pain associated with pancreatic cancer is intense. So if you know of any individuals who've passed from pancreatic cancer, you'll know that of all the forms of cancer, they suffer the most and they are always in constant pain. And the only thing that you can do is to actually block some of the ganglion nerve fibers and to surgically resect some of them so that they don't feel pain from that region of the body. And so, again, it's one of those things that pancreatic cancer is not something that is trivial. He happened to be really lucky. He decided not to actually follow up on it. Again, just to put it in perspective, to prolong your life, to make sure that you can stay alive a little bit longer, the pancreas is removed in its entirety, in its entirety, and a Whipple procedure is performed where different organs are kind of reassigned and reattached to other organs, as you are seeing here. But again, this whole slide, you don't need to worry about anything from this slide at all for the final exam. So Steve Jobs, really bright individual, really well-connected individual, an individual with a number of resources at his disposal, ultimately decided not to use conventional treatments that would have helped prolong his life. And this is why we're talking about belief systems, placebos, and how your brain actually works. So I want you to understand why some individuals actually take that route. So you might criticize Steve Jobs and you might think, well, he had all of those options, why didn't he take them? And sometimes when you're looking at why individuals decide to take non-traditional forms of therapy, even though they might be useful in the case of pancreatic cancer, we don't have a treatment right now for multiple sclerosis. And again, this goes back to Ben Barres. He had sort of researched the oligodendrocytes. Where do these oligodendrocytes that myelinate the nervous system come from? Discovered these type 2A progenitors. And we know, for example, that multiple sclerosis is a disease of the CNS. We get this demyelination. And depending on what areas of the brain or what areas of the CNS get demyelinated, it can cause all sorts of different types of CNS dysfunction. So it could happen, for example, in your visual system. You may have optic neuritis. You may have nystagmus, or the rapid movement of your eyes. Or you could have things that, again, cognitively you're not quite as quick as you used to be. You might have more fatigue. You might actually have a mood disorder that's associated with multiple sclerosis, etc. But for the most part, many of you will associate multiple sclerosis with impairments in sensation or movement. And most often, that's how it gets presented. Again, the diagnosis, because depending on where it's hitting you at any given point in time, can actually be very, very difficult. You actually have to have a number of neurological tests that are done. You have to have a lumbar puncture, so you have to go in to the cerebral spinal fluid. You have to see if there are elevations in certain proteins and certain antibodies. You have to have an evoked potential where you have little electrodes placed into your skin to stimulate different nerve fibers. And you might have MRIs done as well to see if there are any changes within your brain. But it is a very difficult disease to be able to diagnose. And again, you don't have to worry about this slide either. It's mainly just to set the context. Multiple sclerosis affects a lot of Canadians. In Canada, this is particularly true. It affects women much more than it does men. And it's usually in younger individuals than it is in older individuals. And I don't want to kind of sidetrack our discussion about this, but for the most part, you might think, you know, it's not that bad a disorder because it can be the relapsing remitting type. But if you talk to someone who actually has multiple sclerosis, they will tell you it's a very, very debilitating disorder. They would do anything not to have the diagnosis of multiple sclerosis because their lifestyle is impacted. They're not sure when they're stressed out, the next flare-up might be occurring. And in fact, even though the vast majority, 80% of individuals that have a diagnosis of multiple sclerosis have the relapsing remitting type, so you get better, and then suddenly you'll have a bout of multiple sclerosis. You might be not able to see one day, or you might not be able to talk well, you might not be able to swallow, but then it gets better. And you might think, okay, well, that's not so bad. But for the individual that's experiencing this, and these symptoms don't always happen in the same place, it is incredibly challenging for them in terms of their lifestyle. So if you talk to those individuals, they would do anything not to have multiple sclerosis. So why do really smart people decide on taking different types of therapies? Well, one reason, we don't have a good therapy for multiple sclerosis. So even if it's unconventional, you will do anything to be able to avoid getting multiple sclerosis symptoms. One of the reasons is precisely for that. So I think I'm going to stop here for the time being. We'll take a quick break. We'll come back and I'll talk a little bit more about this, and then we'll get into placebo effects, and then we'll start talking about how we're going to be wrapping up the course. Okay, if you don't mind, we'll just continue on. Again, not to get too deep into all of these different types of details, but one of the things that we are more and more sure of is that multiple sclerosis is likely an autoimmune disorder. So one of the reasons why you get a lumbar puncture is to try to determine if, for example, you see an increase in certain types of antibodies and if any of those antibodies are autoreactive antibodies. And one of the things that we think happens is that your own immune cells will actually start attacking these myelin sheaths for a variety of different reasons, causing these inflammatory patches to exist, which means that the action potential coming from whatever neurons that are being affected and being demyelinated will actually be disrupted. So if it's in the optic system or it's part of the optic nerve, you may affect visual processing, etc. If it's involved in controlling the muscles that allow you to swallow or have speech, then those will obviously be affected as well. So because a lot of these are relapsing and remitting forms, even if you have these different types of patches, they can repair themselves. And one of the things that we know from Ben Barres' work is that we can have migration of these type O2A and other glial progenitor cells to these areas of demyelination to actually help in that process. And again, it's just for some background to kind of fill in some of the gaps. You don't need to memorize anything from this particular diagram. Of course, the worst case scenario is if you have what's known as the progressive form of multiple sclerosis, where these patches don't disappear. In fact, they grow in size and number, and these patches might even be evident on an MRI as these white matter hyperintensities that you are seeing here. This is a very abnormal MRI, and you can actually go and pick out the different regions of the brain that would be affected, and as a result, the different types of things that would be affected in those individuals. Again, there is no cure for MS. There are things that can help you manage the inflammation. There are things that could manage the immune system a little bit, so things that are involved in interferon beta or other types of interferons. These drugs can be used and administered to help modulate the immune system, but by themselves, they're not a cure for multiple sclerosis. If you start having a relapsing remitting bout of multiple sclerosis, they may shorten the time that this occurs, but it won't actually prevent it from occurring down the road. And these are not cheap drugs either, so it is a really impactful disease on Canadian healthcare. Again, corticosteroids, because they work in an anti-inflammatory pathway, can also help to reduce the time of relapse. Often there are inflammatory cells that are accumulating, not just immune cells, and as a result, it can help to reduce the time of relapse as well. Most of these drugs that are also prescribed are there to help with the treatment of symptoms. They are not able to reverse nor to prevent multiple sclerosis. And again, it's a very debilitating disorder. Imagine waking up tomorrow not being able to see, swallow, speak, not being able to think clearly, and never knowing when the next bout of that's going to happen or how long that's going to last for you. So it is much more debilitating than we give credit for. And one of the things that, again, I want us to think a little bit about, because this course is also about you being critical thinkers. And I don't know if you have heard of Dr. Zamboni. For some reason, we talk a lot about these really controversial figures. And one of the things that he was interested in, so he's a surgeon and professor at the University of Ferrara. He had a wife who at the time was 37 years old. So in that age range, remember 15 to 40, the age range where multiple sclerosis is most likely to exhibit itself. And she starts to develop the classic symptoms of multiple sclerosis. So she has weakness in one of her arms, which then spreads to weakness on one side of her body. She is confined to a wheelchair for a long period of time. He's a professor, he's a surgeon, he wants to help his wife. And he starts looking in the literature. And in the literature, when he starts reviewing it, some of the research actually stands out for him because of his clinical expertise that says that there's a link between the amount of iron you have in your blood as well as the symptoms of multiple sclerosis. So the literature that he was reading suggested that there was a strong link between iron deposition, the blood vessels that drained the brain, and eventually the cell losses that accompanied the lack of proper drainage from the brain. So the longer your blood is pooled up in your brain, the more iron it deposits there, the more iron deposition leading to more cell death. In his hands, and again, this has not been verified, in fact, it was only in his hands, using ultrasound, he was able to show that of the patients he examined, that 90% of the patients that he examined had abnormal blood vessels, particularly the drainage vessels, the veins that drained the brain. So again, because people had no better options, there was no cure, and he had this really incidental finding, he wanted to know if by opening up these veins, so these abnormal veins happen to be abnormally small or abnormally kinked to prevent blood flow or blood drainage. If you open up these veins, you put a catheter, you take a balloon, you expand these veins, and then you allow these veins to become bigger in diameter, will that help in multiple sclerosis? So he develops what is known as this chronic cerebral insufficiency technique, which he calls the liberation technique. And for these individuals, about 73% of them did not have any of the signs and symptoms of multiple sclerosis. So, of course, immediately people wanted to know if this was a potential and a viable option for them for multiple sclerosis. And because there's no cure, and now he's suddenly suggesting, I have a cure, not just treatment, but a cure for the disorder, of course, people jumped on this right away. Do you think he's right? Because everything we've talked about in this course, I always start that question off that way, and it turns out that they're wrong, right? So this is still debated. It's not really clear. And I think that, again, you don't need to know all of the details here. In one of those types of studies that was done, and this actually affected us here in Canada because it was not a prescribed procedure. A number of Canadians still go down to the Caribbean to have this procedure done. It is not done in the proper way down in the Caribbean as it is in Canada. Canadians have a very high incidence, by the way, of multiple sclerosis. In Saskatchewan, the incidence is very, very high. And because it's so debilitating, a lot of individuals need to have some kind of treatment in order to live their lives normally. And this quickly became what is known as a complementary alternative form of medicine. It kind of looks like medicine, but it's not really medicine. And one of the reasons why is when a University of Buffalo study wanted to know, is this really all mediated? Do those 73% of those individuals that feel better after getting this liberation technique, are they actually better, or do they just feel like they're better? Do they no longer have multiple sclerosis, or do they no longer feel they have multiple sclerosis when in fact they do? So when the University of Buffalo decided to do this randomized control study using controlled individuals where they had everything kind of done up to the part where their veins were expanded, so they underwent anesthesia, they didn't know if they had the treatment or not, and then they were asked, it turns out all of the findings turned out to be a placebo effect. They just felt better, and so it wasn't a real effect in terms of no longer having remission, no longer having a relapse of multiple sclerosis, etc. And unfortunately, because of the way that this works, it's not paid by the Canadian government, it's a private healthcare cost. Mahir Mostich a number of years ago actually went down. He passed away because the surgery was performed inappropriately, and as a Canadian citizen, it caused a great controversy. Should we be supporting these types of complementary and alternative forms of medicine? And again, you might think, why not try something that's more biological? So we're missing these oligodendrocytes. Something happens, the autoimmune disorder causes antibodies to attack, destroy these myelin sheaths. Let's use what we would traditionally use in terms of neurobiology. So one of the things that is used are stem cells, right? So if we're missing these things, we talked about O2A progenitor cells, why not introduce stem cells to kind of replace some of these damaged oligodendrocytes? So this was sort of a different approach to this. And one of the things that is sort of interesting about this particular study that was done, it's now in a larger sort of trial. At the University of Genoa, instead of trying to replace the damaged oligodendrocytes by giving O2A progenitors, which is what Ben Barres and other individuals had suggested doing, it is still a therapy that is being pursued in the United States. But in Genoa, they decided to take a different approach. In one of the earlier slides, I told you that this is an autoimmune disorder. And one of the things that they wanted to know is if we could restart the immune system so that it's not producing these autoantibodies, can we actually get these individuals to feel better? And one of the drugs that is typically given is mitoxantrone. And as a control, there were some patients that received the drug mitoxantrone, and other individuals had stem cells that were harvested from their bone marrow and then reintroduced after treatment with an immune suppressant. And when they did this, and the cells went back and formed the normal immune system again after being immunosuppressed, when they examined the endpoint on an MRI, so they were looking at T2-weighted lesions on an MRI, so those little white hyperintensities that I showed you earlier, and they were using gadolinium sort of as a contrast medium. So the white matter hyperintensities are really hard to pick up. We have to inject with gadolinium as a contrast material. It stains those hyperintensities. And as a objective measure, if we can restart the immune system, so we take out your bone marrow stem cells, we treat them with immunosuppressants, we reintroduce them back into you, the individuals that had their bone marrow stem cells treated, suppressed, and therefore had their immune system suppressed, actually had fewer lesions in an objective measure. So here is not just asking, are you feeling better? Do you have less lesions that we can measure on an MRI? It turns out that this actually seems to work, and it also helps with their symptoms. So there are some really exciting things coming up, but I wanted you to be aware of them because sometimes when you hear about these exciting findings that sound like they're really based on science. So the liberation technique and the therapy, if you were to just read through Dr. Zamboni's articles, you would think, wow, it makes perfect sense. There is a scientific, logical rationale for why this should work. Iron deposition, increased oxidative radical formation, cells dying, causing the formation of multiple sclerosis. It makes sense, but in fact, it's not properly controlled, and most of it turns out to be a placebo effect. Do you have any questions on this before we go on? And again, just to set up the placebo effect, which we're talking about next. Any questions? Okay. How many of you have heard of the placebo effect? How many of you think the placebo effect is real? How many of you think you're going to do really well on your final exam? Obviously, you don't believe in the placebo effect. Okay, so we'll see if I can introduce a placebo effect for you. And again, you don't have to worry about these next few slides. It's just to give you some context for this. A lot of this was thought to be not good. Like the placebo effect for a very long period of time was thought to be sort of magic, in fact. The person who actually came up with the basis for the placebo effect is known as Franz Anton Mesmer from mesmerism. And one of the things that he suggested that he was able to do is that he could pass on this effect, a force of nature, which he called animal magnetism. You could actually touch something that's inanimate, like a piece of wood. I can imbue this wood, because I have the animal magnetism, with my power. And by doing so, this wood would then have this animal magnetism associated with it. And this force of nature, a new force of nature beyond the ones that had been known in the 18th century, he said that if you could touch this imbued piece of wood, that it could evoke different types of emotional responses, you could be healed, you could do a lot of amazing things through this animal magnetism. it had gotten to a point almost like what we would do now, where, for example, you hear that a technique or a treatment is really, really powerful. You want to test to see if that actually is true or those claims are valid. And that's the type of way in which science is working. One of the ways in which this has sort of been passed on to us is from Louis XIV. He actually hears about this animal magnetism. He sees evidence that animal magnetism is actually real, doesn't have any reason to doubt that it's real, but he wants to know, do scientists think that this is real? So he appoints a royal commission, similar to what we would do today, and he hires the sort of all-star team of the best scientists that were around at the time in the 18th century. So Benjamin Franklin from the United States, and then Antoine Lavoisier from France. They're commissioned together, find out if animal magnetism is real or if it's completely fake and he's just making all of this stuff up. And one of the things that they decided to do was actually develop the very first controlled experiments. So this is why we do controlled experiments today. So they decided that, hey, we need to know if animal magnetism exists or doesn't exist. And so first, they actually followed Franz Anton Mesmer. They saw what he did as he touched different trees in the woods, imbuing them with animal magnetism, and then telling individuals to go and hug that tree. When you hug that tree, you're going to feel like my power pass into you, and then you're going to have you're going to be healed, you're going to be having these emotional responses. So they're watching all of this, and they see all of this happening. So they actually go in, see and observe exactly what he does, and then they see that indeed there's some kind of magical force. The trees that he touched, when these other individuals touch them, they have fainting spells, they feel like they've been cured of disease. Everything seems to work exactly as he has said. What would you do that's different as scientists in the 21st century? You wouldn't have watched it, no? What would you have done differently? Anything? Yes? You wouldn't let the participants watch. And one of the things that they did was to actually set up what is known as the first control experiment. So they did the same experiments, and they actually went in and they touched random trees, or they had Franz Anton Mesmer touch random trees, but they didn't actually let the participants know which trees they had touched. So when they started to have these emotional responses, they actually told them after, that's not the tree that he touched. And so at the end of all of this, though, this is really the important key message. They realized that these emotional responses and the good effects that they were observing, these effects were due to the imagination. But they didn't discount the fact that there were real effects that they were observing. So people really were crying. People really were fainting. They weren't pretending to faint. They were doing so because they believed that what they were touching actually was imbued with this form of animal magnetism. So this gets into our concept now of the placebo. And for the very longest period of time, people knew that there had to be something that we could measure in terms of these imagination-based effects. So 1955 is the first sort of place in which the experiments that were done on the placebo were actually published in a peer-reviewed journal. And this was Henry Beecher. And one of the things that he decided to do was to use what is known now as a double-blind procedure. So neither the patients that were being treated nor the physicians knew who had the real drug and who had the placebo drug. And one of the things that he found that in his study, and he was again primarily interested in this because of the Korean War, so this is 1955. At the end of the Korean War, a number of surgeons indicated that, you know, I actually don't have to give my patients morphine to make them feel like they are not feeling pain anymore. I just have to tell them I'm giving morphine and they feel better. And he decided to test this at the end of the Korean War when it finished. And he started to realize very, very quickly, this is actually a real effect. As long as the patients believe that I'm giving them morphine and I'm injecting them with morphine, some of them are actually going to feel less pain. They're not going to be staying up. They're going to be able to go to sleep. I can measure all of these effects. And these follow-up studies actually said placebo is a real effect. It is a real neurobiological effect, and that's what I want us to concentrate on over the next lecture and a half. And in fact, placebo works really well on some parts of the nervous system. So for example, if you have a psychiatric condition, certain types of psychiatric conditions like mood disorders, depression being one of them, or anxiety, if you give a placebo, it works almost as well as the drug does. So again, placebos have been shown in the literature to be very, very powerful for psychiatric disorders. It is incredibly good for analgesia. So placebos work on moderating pain and modulating pain responses. Surprisingly, or maybe not surprisingly, it also works really well on Parkinson's disease. You can tell someone with Parkinson's disease that you're giving them levodopa. They actually move better when you tell them that you're getting this drug, and it turns out it's a placebo. You don't let them know, but as long as a Parkinson's patient believes that they're getting treated, they actually will respond better in terms of their motor movement. And some of the best research was actually performed by Kirsch and Saperstein, so I'll mention them several times. And again, different things for depression, antidepressant medication, giving them a placebo works just as well as antidepressants. And again, there's a lot of significant research that's actually being done suggesting that the placebo effect, which we don't understand in 2018, is actually increasing. So if you happen to read about Hedges G and different published trials for the antidepressants, one of the things that's surprising to learn is that antidepressant medication in 2018 is not as good as antidepressant medication in 1988. Can you think of a reason why? It's all due to the placebo effect. Who do I compare them to? Randomized controlled placebo patients. The placebo group, when you give depressed individuals placebo, they are responding much, much better now than they did before. That is not understood. We don't know why in 2018 the placebo group actually responds at a higher level than they did back in 1988. So when you compare against placebo and you look at the overall efficacy of antidepressant medication, it's been steady. The placebo group has been catching up much faster than they did back in 1988 or 1998. So there are various factors that are thought to underlie the placebo. There are psychosocial factors. You have to think that you're in the right setting. You have to also have the sort of right delivery of this, et cetera. And I'm going to go through this with you. And it also requires a little bit of neurobiology. It requires some conditioning. It requires some patient-physician interactions. And I'm going to try to prove this to you in the next little while. So let's go back to how we would do this now. In 1955, Henry Beecher published in Lancet one of the first studies that looked at placebo effects on pain and analgesia. I told you earlier on that of all the things that you can examine, analgesia, or the control of the sensation of pain, is one of those things that placebo works on really well. How would we do this in 2018 versus 1955? So one of the things that I told you is that there's some context. You need to feel like you're being given medication. So how do we test this, and how do we determine if there is a psychosocial element or if it's just due to some other biological factor? So I don't know if you have ever undertaken a psychology study or if you have ever done a visual analog scale. So this is a visual analog scale from 0 to 10. And you are asked, on this particular scale right now, when you're in the hospital, tell me on this scale how much pain you are feeling. So you might indicate, for example, oops, I'm hoping it works. I'm feeling this level of pain. Like it's really high. Like I'm feeling like 10 being like the absolute worst pain I've ever had in my life. Right now I'm feeling like it's about an 8. And you go and measure this, and then you want to know, can I give a drug that reduces on this visual analog scale for pain? Can I give you a drug or a placebo that will reduce the level of pain that you are feeling right now? So this is sort of why we're talking about a visual analog scale, this VAS, that is over here on the bottom. And I might ask you, after giving you a traditional drug like an opiate or some kind of other analgesia drug, So whether it's an opioid or a non-opioid, something to help you with your pain, and then go back and ask you later after you have been given this drug. So I'm your physician, I've just given you this drug, you've taken it, I come back an hour later and then I ask you, how are you feeling now? How much pain are you in? And then you check off, oh, a little less pain, and this is where I'm feeling right now in terms of the visual analog scale. And this is sort of what is being tested here. But we do it a very, very different way now, and this is where we have either hidden application or open application. So this is where a doctor is coming in, checking up on you, doing what I've just done, and then giving you an injection of a drug or giving you a pill or something to treat that level of pain. And this is sort of the open application. Versus a hidden application. I'm giving you the same drug, but you don't know that I'm giving you that drug. It's just you're being given that drug at the same dose and at the same level as you would by this physician. So there's a hidden application versus an open application. And then you are asked to rate your pain either in the open or in the hidden application. And one of the things, again, which is surprising, so keep in mind that these different drugs are all drugs that are used to control your pain level. And what we're seeing is that if you have pain and you are given morphine, which is, again, known to control pain, really bad forms of pain, you will see that your visual analog scale might have started off here, but then it's reduced by like 2 points. And this is when the doctor is giving it to you. How are you feeling today? Are you not? Oh, you're in a lot of pain. Let me give you something for that. And then if I check back on you later, your pain scale has gone down by about 2 points. If I don't do that, though, and I give you exactly the same amount of drug, and I give you exactly the same dosage over the same time course, and then I go back and test on your level of pain, you're still feeling more pain. I'm giving you the same drug, I'm giving you the same dosage, I'm treating you over the same period of time. The only difference is here, there's a psychosocial context for the drug being given. So something else is required for you to get the maximal effects of analgesia. Do you see that before I go on? So these bars are not the same size. The drug itself, although it is an analgesic, I get a better effect of the analgesic when I think the doctor is giving it to me, or when the doctor is giving it to me, I think it's going to help me more than if I get the same dose through a hidden machine. Is that clear for everyone? And it doesn't matter what drug we look at for the treatment of analgesia, so whether it's morphine, the traditional drug, if I give you tramadol, or others, even non-opioids that work on different pain mechanisms, if I give you ketorolac, it doesn't matter how I administer it. In terms of the control of pain, this difference is a placebo effect. You feel better if the doctor is giving it to you versus if you get exactly the same dose of drug over the same course of time, given it to you in a way that you don't see the drug being delivered. So this is that missing gap, which is due to a placebo effect, an enhancement of that drug effect. Any questions on this before we go on? Thanks, Mike. Yes?
Sorry, for the hidden one, you don't see it, but you know you're still being given the drug.
You know you're being given a drug.
Okay.
Yes.
So you can't see it.
And again, you have to keep this in mind. The drug is working. The drug is actually making you feel less pain. So your pain scores are going down. They're just not going down as much as you would if it's being given it to you by a physician. Yes? Yes.
Then they are given a drug and they are suspicious that it may be a placebo. Then would the effect work, less effective on them?
So your colleague was asking an interesting question. So you're a neuroscience student. You know all about the placebo effect. You go into the hospital and you know about the placebo effect. Will you still have the placebo effect? I'm pretty sure you will. Yeah. Yes. I'm not sure. Why are the error bars so big? Again, I'm not sure.
Well, it's just like the other ones are all significant. It seems that way.
Again, this is more of a trend rather than actually measuring statistical significance. Any other questions? Are you going to go test that out? No. Try not to. Try to stay as healthy as you can for as long as you can. All right, so it works really well here. This is for expectation. You're actually expecting a better response if you're getting this from your physician. So how does this potentially work? And it turns out that there are a number of systems that have been well described. And again, a lot of this goes back to Kirsch and Saperstein, who mapped out what areas of the brain might be involved in determining this placebo effect, that enhancement of the effect beyond what we get just from the drug itself. So there are two different systems that are at play, one of these being the dopaminergic system and the other one being the opioidergic system within your brains. These are the two that respond best to placebo. And one of the things that we know is the most powerful area of the brain that involves the dopaminergic system is the VTA to nucleus accumbens. This area here can actually be influenced by the placebo effect. The opioid system is a little bit different. So this is a descending control system. Descending means it starts off in the cerebral cortex and it's going progressively to lower areas of the CNS. So this cerebral cortex, neurons in the cerebral cortex, in the dorsolateral part of the prefrontal cortex, can make synapses onto the periaqueductal gray. So this PAG is the periaqueductal gray. Hopefully you have learned that in one of your physiology courses. That's the cluster of neurons that's involved in trying to synthesize the pain response. It will then signal through the remainder of this descending pain pathway. So the rostral ventral medial pathway, the descending pathway into your spinal cord. And one of the things that we know is if we can alter the synaptic transmission in this descending pain pathway that involves the release of opioids at each of these different synapses, we can actually influence the placebo effect. So we know this because in this particular pathway, and if we go back and examine this particular pathway at any of these different junctions, because again, remember pain pathways are integrated at the level of the spinal cord. If we use different types of drugs like opioid receptor antagonists, so each of these purple boxes represent the opioid pathway. If we use an opioid receptor antagonist like naloxone or naltrexone, these effects that we see here are blocked. We don't see them anymore. So we know that this descending pathway is responsible for the placebo effect associated with pain responses. And this opioidergic pathway is incredibly important. And that's one of the proofs of principle that using opioid receptor antagonists actually reverse the or block the placebo effects that we would see associated with analgesia. The other input to this is these the cholecystokinin neurons, and these CCK-ergic neurons that you see here are really important as well. If we stimulate this group of neurons, this can also block the placebo effect. So if we have, for example, if we can isolate and stimulate the cholecystokinin pathway and the release of cholecystokinin, it can also inhibit the opioid pathway as well. So we need this descending pathway intact for the placebo effect to occur. And it starts off at the dorsolateral prefrontal cortex, and at each of these different synaptic connections, the analgesic effects of placebo can be blocked with naltrexone or naloxone. And I think I'm just going to go through these last few slides related to these SNPs, because you need to know these for the upcoming quiz, and I'll let you know in advance these next few slides are going to be well represented on the upcoming quiz as well as on your final exam. So you were asking a really interesting question earlier, and I have to apologize. I kind of gave you an off-the-cuff response. The real response would be, I'm not sure, because I would have to genotype you. So if you're really interested in that, come back with me. We'll go over to one of the labs over at CAMH. We'll get you genotyped, and then we'll know the real answer. Because some people are actually better responders to placebo than others. So these really have to do with these single nucleotide polymorphisms that are part of our genotype right now. We know, for example, that in addition to the opioid pathway and in addition to the dopamine pathway that are very heavily influencing and very heavily involved in the placebo response, that certain individuals that have genetic variations in the serotonin pathway are also different in terms of their placebo responsiveness. So one of these is tryptophan hydroxylase 2, or TPH2, and this gene promoter region, the promoter region for TPH2 actually has two different polymorphisms. At position 703, you can either have G or a T, so either a glycine or a threonine at that particular variant. If you have two copies of G at position 703, that means that you're more likely to be a better clinical responder to placebos. So we can actually genotype you to see if you're going to respond better to a placebo or not. Earlier on in this course, we talked about the 5-HT-PLLR. Do you remember that at all? We talked about this associated with depression. If you have the long version of the 5-HT-PLLR, the transporter, you're also a better responder for placebos. You're more likely to respond to a placebo than someone who has the short version of the promoter. Okay, so these are two different types of SNPs, or two different genetic variations that are associated with placebo. Why might this be important, do you think? And who might it be important to? Any ideas? But is it important to you? Is it important to someone else? Like, do you need to know if you're going to respond to a placebo?
Oh, no.
No? So who might it be really important to? Yes? Yep.
Would it be for people who are easily addicted?
Easily addicted to pain medication? Okay, potentially. Who else? Why the doctor? They need to know how to treat you, but you have a drug, so either you're going to give them a drug or not. Usually your doctor gives you a drug, not a placebo, I hope. Anyone else? Yes?
I think it would matter to the person who's receiving the treatment.
If the treatment works, great. If the placebo doesn't work, do you really care?
But if you're able to take a placebo instead of a drug,
Is there a reason why you would want a placebo versus a drug?
because it's not making alterations to your system.
No alterations to your system. Okay, potentially. So think like, who has a lot of money? Not Steve Jobs. Okay, we passed that. Who else has a lot of money? Yes. Yeah, so this is actually a really big part of the pharmaceutical industry. Remember I told you earlier that compared to placebos, we always compare the drug versus a placebo. Now, we need to be able to demonstrate that the drug is performing much better than the placebo that they're being given. If we have a lot of people who are placebo responders in our group, that would actually skew our results. So nowadays, the drug companies are actually screening ahead of time to make sure that high-responding placebo responders are taken out of that control group just to give them a better responsiveness. A couple of other genetic markers, again, for you to be aware of as well. So individuals that have higher monoamine oxidase A activity, and this is determined by whether you have a G or a T polymorphism, will have different responsiveness. So if you have the GG variant, so you have you have two copies of glycine on MAOA, this polymorphism actually give you a better placebo response than if you have a G and a T or two T copies of the MAO gene. The same thing for COMT. Again, another polymorphism at position 158. So if you have valine, you have lower levels of COMT activity versus individuals that have the M allele, and which means that lower COMT activity, more neurotransmitters, so you have a higher placebo response. Again, surprisingly, and this goes back to what your colleague was mentioning earlier, a lot of this is also involved in mood disorders as well. And people are trying to make this link with mood disorders. And do placebo responders actually also have like different types of mood disorders? And it turns out that many of these genetic markers that are involved are also really highly represented in things like social anxiety as well as in major depression. Not surprising, because this is where we're looking at the breakdown of neurotransmitters, the synthesis of noradrenaline, and others. And again, just as an overview for you, please do not memorize this. You don't need to memorize any of the things that you're seeing here. It's just to highlight TPH2 is this pathway that I showed you earlier. So this involves eventually the formation of 5-HT, and MAO is sort of used in the breakdown of various neurotransmitters. So it does directly influence the neurotransmitter system. And again, this is not a slide that you need to memorize any of the details for. It's just to help you visualize like what some of these different systems are or what TPH2 is doing, but I'm not going to ask you any questions related to this slide or any of the details that are on this slide. And if it's okay, I have 5 minutes. Just so that we don't have to continue this on into the next lecture, I'm just going to go through these last 2 slides. The other things that I want you to be aware of as well, that descending pathway that I showed you earlier started off in the cortex. The dorsolateral prefrontal cortex is particularly affected in Alzheimer's disease. In Alzheimer's disease patients, when you go in and you test them, and they have reductions in what is known as the frontal assessment battery test. If you go in and you test them this way, so let's say it's 2018, I go in and I score relatively high on my frontal assessment battery test, which shows me that my frontal cortex is working well. Today, in 2018, I will have a pretty good placebo response because my dorsolateral prefrontal cortex is intact. Let's assume for the time being all my other genetic markers are neutral, and I'm a pretty good placebo responder. However, let's say I have Alzheimer's, this area of my brain is degenerating, I go back in, my frontal assessment battery test in 2019 is lower. I will also see if I were tested for placebo responsiveness in 2019 as my frontal cortex degenerates, as that dorsolateral prefrontal cortex degenerates, that opioid descending pathway, I will also have a lower placebo response. So again, another piece of evidence to suggest that that pathway I showed you earlier is really important. So again, we've already talked about naloxone, but we can also do this non-pharmacologically. So you might wonder, well, you know, we're using naloxone, which is an opioid receptor antagonist. Can we do this in other ways? And one of the other ways that you can do this is by using TMS. So if I could find an area of the brain that is involved in activation and the release of opioid, and the release of opioid is associated with the placebo effect, and it's continuing down this chain, if I could somehow block that chain from occurring and block the release of opioid, would I be able to show that now the placebo effect is being reduced? So how many of you have heard of TMS, transcranial magnetic stimulation? Most of you have. What target would you pick to block the release of opioid in terms of taking a large magnet and stimulating or blocking that area of the brain? What would you pick? Because you have a number of targets. You could pick things in the spinal cord, way back here. You could pick other areas like the DVM. What would you pick? What's an easy target for TMS? Any guesses? Any guesses? No guesses. Sorry? It's done on your head. What region would you pick? I would also pick the dorsolateral prefrontal cortex. High five. Awesome. So by putting and inhibiting the dorsolateral prefrontal cortex, you can actually block placebos that are involved in analgesia. They don't feel the placebo effect anymore, etc. So again, suggesting that this descending pathway is incredibly important. In our next lecture, I want you to think a little bit about this over the weekend. Do animals actually experience placebo? So this involves the dorsolateral prefrontal cortex stimulating and getting activated and turning on the release of opioids. Can we do the same thing and can we show the same thing in animal models? How many of you say, yes, we can? How many of you say, no, we can't? How many of you have arms? Okay, that's what I thought. Awesome. So I'm going to try to show you in the next lecture, which you will have on your final exam as well, how these animal studies in placebo work. And then we'll kind of wrap things up on things related to meditation. And if any of you are interested in talking to your TAs about any of the sort of written summary, Jessica and James are here, and I'll also stick around to answer any of the questions that you have. And again, just as a reminder, the deadline is pushed to December the 7th, which is the Friday, for 12 noon. I know some of you have been asking for it. I'll post that officially on the announcements page as well. Have a good weekend. I'll see you all next week.